Hello, everyone, and welcome to another National Geographic Learning Google Hangout. My name is Samantha Zolke, and I am joining you from Washington, D.C., where National Geographic headquarters are located. But we won't stay here for long, because today we're traveling to Panama in Central America with my friend Clare Fieseler and Dr. Carl Castillo. Clare has been joining us for the past few Hangouts from research stations in the Bahamas, Belize, and most recently Miami, Florida. But today, she and Carl are speaking to us from our host, the Smithsonian's Tropical Research Institute in Panama. Hi, Clare and Carl. How are you guys today?
Good, good. How are you doing this morning, Sam?
Good. It looks beautiful there.
Here in Panama, we're at a place called Bocas del Toro, which is on La Isla de Colón, which is an island here in Panama. And we're at this beautiful research station that is, it's run and managed by the Smithsonian, which is based out of Washington, D.C. as well, just like National Geographic. It is, we've got some tropical vegetation. You can see the lake behind us. We're just going to pan out here a little bit, see the beginning of the station, and it's a real tour inside. Can you still see us okay, Sam?
We can see you. You're freezing up a little bit. But before we get to the tour, I want to tell our viewers how they can ask you guys questions and be active participants in our Hangout today. We are going to be monitoring our Google Hangout page and the National Geographic Learning Twitter feed. So as you're watching, if you have questions, we want you guys to tweet at us at ExploreInside, that's INSIDE, or post your questions on the Hangout page. And I think what we want to do is start broad and see where you guys are at in the world. So I'm going to queue up a map to show us a little bit of where we are.
All right, and we're going to go into the lab while you explain that.
Awesome. So I'm going to share a map from our National Geographic Google Hangout, or our National Geographic MapMaker Interactive. I want to make sure that everyone can see that. Oops. Which you can access by going to natgeoed.org. So you'll see we have pink markers where I am in Washington, D.C., and in Panama, where Clare and Carl are. And the blue circles represent past Hangouts. So we mentioned earlier that Clare talked to us earlier from Belize, from the Bahamas, and from Miami, Florida. And so what we want to do today is explore a little bit of the geography of these areas. You'll notice we have our equator here. And as we all know, the equator is the imaginary line that separates the northern hemisphere from the southern hemisphere. And so Clare and Carl are going to talk a little bit about the big picture stuff as they talk about their research. And so what we want you guys to keep in mind today as you're watching is how the environment affects what they're doing in their work and the questions that they're trying to solve as explorers. And so if we zoom out here, we can see that the areas that they're all studying are our equatorial climate regions. And we know this because we've turned on a layer about our climate zones. We can zoom in and see that most of our study areas are in our humid equatorial climate. Now, if we want to learn a little bit more about the specific weather there, we can turn on our surface temperatures. And I can see the interaction between these two things by turning these off and on. Oops. Oh, it doesn't seem to be working. So let me try a different one. Zoom out so we can see all of our area. And again today, as you're watching this Hangout, what we want you to keep in mind is how the environment impacts the study questions that Clare and Carl are working on. So we can see some patterns here, but we'll come back to these later. So I'm going to end my screen share now and turn things over to Claire to ask, to introduce herself, tell us a little bit more about herself as a Young Explorer grantee, and to introduce Dr. Castillo.
Great. Well, thanks, Sam. Thanks for that great introduction. It's always great to be in the tropics, like you said. Just so much interesting questions because we're in such a unique environment that's so different from what we experience in the United States. As Sam said, my name is Clare Fieseler. I'm a National Geographic Young Explorer, and I travel around the Caribbean looking at climate change and how coral reefs, which are a really important ecosystem in the tropics, how they're responding to how the ocean's getting warmer and warmer. Now, my research is very much aligned with Dr. Carl Castillo's work. He works on a lot of different questions. He's been doing this for a very long time, and he actually grew up in the Caribbean region. And he's a professor at UNC Chapel Hill. He's actually my research advisor. I learned so much from him. And I'm so excited to share his research, which is really interesting about the way we look back into the past, of course, the future. So, Carl?
Yeah, so again, my name is Carl Castillo. I'm originally from Belize, actually. Did much of my undergraduate work in the U.S., Southeast Missouri State University, at the University of Southern to UNC Chapel Hill as a professor as well. So welcome, everyone. I'm very excited to share my work and the interesting things we're doing down here in Panama.
Great. So Sam, where do you want to start?
I'd love to learn a little bit more about the research station that you're at now that you're inside. Before we got to see some of the beautiful vegetation, and I was listening to hear some of those tropical noises we heard the other day when we were chatting with you guys. But now that you're inside, could you tell us a little bit more about where you're at and the role that the Smithsonian plays there?
Absolutely. So the Smithsonian has run this research station since I think the 1960s, and they have many different research stations throughout the Caribbean. And here at the Pacific Research Station, because we're on an island, researchers can study both the land and the ocean. Use of boats here, and the reefs are just offshore. We actually were on a boat yesterday. Carl and I were doing work. I'll show you some videos from it, and it was just a 10-minute boat ride off the dock from the station here, and so it's really easy for researchers to come down. And the Smithsonian, it's mostly just for scientists, but there are some school groups that come down. Panama is really interesting because it's where the two continents kind of come together, South America and North America, so there's a lot of biodiversity here. And that's one of the key things that's so important in studying at the Smithsonian Tropical Research Institute. But I think we have a video that might kind of give a better sense of why this environment is so important and why it's so many people come to study it. So should we cue that video?
Yeah, that would be great. And while we're queuing that up, I want to remind everyone that Panama is an isthmus, which is one of my favorite geography words because it's so fun to say.
What is really cool here is that we also have, lab facilities. I think the Smithsonian is properly outfitted for researchers in terms of having a, suitable mess hall, dormitories, and everything that researchers actually need to get their work done.
Yes.
Is the food good?
Everything here, we sleep.
So yeah, the food is great. I can tell you.
So yeah. We know that's the most important thing.
It's so really good.
All right, I think we have that video almost ready.
Great. And one of the other things I should mention is that we bring all of our own gear. So we brought 20 different giant boxes of gear, and I'll maybe just show this to you. These boxes right here, about 20 of them to bring all of our gear. It's a lot of work to bring 20 huge boxes, and kind of really heavy. But we need to research, but it's also to make sure we have really fun, have all the right equipment to do the work that we need to do.
How much, Claire? How much do each of those boxes weigh? Do you have any idea?
they're about 50 pounds each, so it takes a lot to get them to and fro. They are, and so not only are we doing research, but we're doing a lot of figuring out how to get everything from point A to point B in a really different environment and climate.
Excellent. And is this?
So I think we're going to try to do the video from the Smithsonian. Go ahead.
I think we're having a little bit of difficulty getting it to play. And so if we can't get it to play, we'll post it to our Google Hangout page later so that everyone can see it. We'll give it another minute, but in the meantime, I'll ask one more question. Is this each of yours first trip to the Smithsonian Research Station in Panama, or is this your first time in Panama?
So this is both my first time in Panama and my first time to the Smithsonian Research Institute. I've actually been to the Smithsonian Research Facility in Belize, but again, this is just my first trip down to Panama and to the Institute here.
So everything is really new for us, and that's what's so exciting. We had our very first dive yesterday, and we'll show you a video of that a little bit later. So it's always interesting because as Sam said, we have different latitudes below the equator. All reefs are different, so the reefs in Belize are going to look a little different from the reefs in Panama. So we were exploring yesterday and seeing what those differences are like. Yeah.
Great. Well, I'm not entirely sure that we're going to be able to get that video to show. So I think what we'll do is we promise we'll post it to our Twitter feed later and our Google Hangout page because it is great. So we're just going to have to make sure that you guys keep coming back for more by holding that a little bit ransom. And move on to, so Claire and Carl, what are you guys doing down there? You say it's your first time in Panama. You're at this exciting research station with excellent food. You hear birds every day. Why are you there?
So, so the central question that we're interested in is we're trying to understand how coral reefs are responding to climate change. We as you showed earlier in your map, we're interested primarily for coral species over a particular geographic range that extends from the northern extreme in Florida all the way to the southern extreme of these. Interested in the.
We can bring that up real quick while Carl explains the rest.
So the starlet coral, which scientific name Siderastra siderea, I love it, so I always say the scientific name. And then we're also interested in a brain coral, which is Pseudodichlora strigosa, again, scientific name. And again, the geographic range of both species extends from Florida all the way down to a little bit beyond where Panama is located. And this is why we're interested in species along this entire geographic range. Again, the primary question here is how are corals responding to climate change, and how does their environmental history, what they're exposed to previously, determine how they will likely respond to previous changes and seawater temperature or climate change overall.
So Carl, how can you look at the history? So you said the history of the coral is really important to understand what's been happening in the past, what's been happening in the future. So how do you understand what sort of environmental history a coral animal in the ocean has experienced? Do you look at it? What sort of methods do we use?
Yeah. So
real quickly, do we have that photo, Sam, of our study species?
I'm looking into it. I'm trying to see it, but I can't find it right now. But a quick question while I check that out. So if I understand correctly, and I want to make sure our students understand at home, the reason that we're in the equator right now is because these coral species that you're studying, this is where they live?
Yes. So again, we find these two particular species from their southern extreme region, Ecuador area, all the way up to towards Florida. So this is one of the reasons why we're working in this particular region, and to try to see over which corals are responding to climate change.
Okay.
Excellent.
So, Carl, do you want to, maybe we can show everybody how we go about sampling and doing research on the corals that we find here?
Sure.
Okay.
Let's go ahead and do that.
All right, cool. So we're just going to go out on the balcony a little bit. Sam, can you still see us okay?
I can. I think you might have to move a little slowly and talk a little slowly outside.
Okay.
Good.
Yeah. So what Carl has here is actually a scuba tank. And what's inside of that scuba tank is compressed air. And we use compressed air to breathe underwater. But another thing we can use it for is to power something called a pneumatic drill. And Carl, do you want to demonstrate how we do this?
One of the questions that Claire asked earlier is, how are we able to get some information about the environmental history that corals are exposed to? Well, you could pretty much see what the temperature, environmental parameter you're interested in. But you can also collect what we call a coral core. So this is a coral core that we actually extracted yesterday. And we can use this as a way to try to get some idea of, how corals are responding to the environment that they're exposed to.
Can I see this a little bit, Carl? So can you guys see this okay?
Yeah, that looks
on
great.
Top,
that is a huge
on
core.
Top, yeah, it's actually one of the smaller ones we have. On the top, you can see this dark coloration, and that's actually the live coral tissue. And the bottom here is all the skeleton. So all that skeleton has grown over the past 50, 60 years.
And again, you can use that. Go ahead, Sam.
How big are these corals that you're extracting these cores from? Can you? So, are they like?
It ranges from over a meter in height to smaller ones. This one is I guess approximately 30 centimeters. So again, it could be over a meter all the way down to 30 centimeters. Use that to get some information about the environmental history of the coral.
But some of these corals are the size of a bathtub. They can be really big.
Yeah.
Yeah.
Wow.
So again, go ahead.
Oh no, I was just
sorry. I think we're experiencing a delay.
These corals are much bigger than I expected them to be. When I think of corals, I think of Finding Nemo little corals, and these are huge.
So before we move on into talking a little bit about what information we're able to draw from this, maybe we could demonstrate how this is actually extracted and collected, so that our viewers can get some idea of what's going on in terms of collecting the coral. So we use what I have here is a pneumatic drill, and essentially this means that it's powered by some type of air supply, and we use this device to extract our coral core. You could see that it's connected to a regular scuba tank. So this is our air source that we use. So I'll go ahead and just connect this and maybe give it. Here is our drill bit. This is the drill bit we actually use to extract the coral core. This is connected to the drill. And you could see that the drill is connected by an air hose to our scuba tank. So if I go ahead and turn the air on device to pretty much extract our coral core. So you could see that. And again, this is, I guess, maybe about a foot and a half in length. And if we're interested in getting longer cores, then we use what we call an extension rod.
So for those big cores that are the big corals that are the size of bathtubs, Carl makes the drill bit much longer, and allows us to get cores that are as long as a yardstick sometimes. And actually, Sam, I think there's a video that Carl and I took yesterday of him doing this underwater. So maybe if we can try to cue that up, and it's underwater, which is pretty cool. Carl, why don't we take the core and go on inside? Sure.
Yeah, we'll cue that up while you guys are in transit.
And again, this was something we extracted yesterday, so this is brand new information. And we're gonna about 80 of these, right, Carl?
Yes.
We'll collect about 80 of these from across Panama. And when we were in Florida a couple weeks ago, we collected them. It takes a long time. We can only collect about 10 a day because the process of getting out to the reef and extracting them is really time-consuming. So this is really, really valuable to us. It takes a lot of money and a lot of manpower. We've got a 7-person team just to get it done.
Wow. About how long does it take to extract one core?
So, interestingly, we use either a hydraulic system, which means it's powered by some type of fluid source, or that pneumatic system that I just demonstrated. Pneumatic or the air system, it takes about 30 to 45 minutes per core. If you're using the hydraulic system, which is the fluid filled, so pretty much fluid being in the house, it takes about a little less than 30 minutes or so per core.
Yeah.
Awesome. That is crazy. So we're still,
so
we're having, go ahead, Claire.
I was going to say, you know, if the video is going to be ready, we would show it. One of the questions we get a lot is, does the coring hurt the coral? And so, Carl, do you want to answer that question?
So, essentially, when a core is extracted, the only live part is the top part of the core. Let me try to get that shown there. So this is where the only section of the particular coral colony, or the coral, that is live when the core is extracted. The additional part is, you know, the growth of the coral that was deposited from year to year as the coral is growing. And so this is the information that we're trying to use to get some idea of, you know, changes in coral growth that occurred in the environmental conditions that the corals are exposed to.
So that core from the colony, then we can't just leave a hole in the colony. So what we do is we try to plug it with something that is similar. So because coral skeleton is really dense, we want to fill it with something. This is made out of cement or concrete. It takes a long time to make these, the exact shape of the actual core. But we end up, what we do is we hammer this, just like this, to the colony, to the core, or fit the core hole, and cause any sort
additional
of
damage
additional damage. And so, you know, it nails, yeah, it does have an impact, but it's not going to kill the colony. And we think our methods are pretty good, and we always want to make sure that we're making as little impact as possible. So that's one of the ways we do it. And I can also show you some photos of how we monitor the, you know, to make sure that our drilling process doesn't hurt the core, right, the colony. And
we're able to return, you know, to every single colony that we cored from year to year, and, you know, kind of try to follow to see, you know, if there's any particular impact on that colony. And Claire does a great job photo documenting, you know, every single colony pre and post core extraction, which I think helps a lot. And again, we're particularly interested are okay after we've extracted our particular coral.
Well, I think
that's a lot of information.
It is a lot of information, and I can't even really tell the difference between the fake core and the real core. But I think we have that video ready that shows the extraction process. So I'm going to try and pull that up for us quickly.
Great. And Sam, the last 30 seconds is really when you can see the process the best.
Okay, great. All right. How's that look? You see it?
That looks great.
All right, so I'm going to play this.
That's Carl right there, and I'm filming him underwater, and we're in about 15 feet of water. And as you can see, Carl's holding this the big drill. It's much bigger than the drill we just showed you. And he's standing over a colony, a coral colony. It looks a little like a rock, but it is a live animal, many live animals. And what he's trying to do is get in a good position so that he can get the drill vertical so it's not shaking around and creating any damage. You just want to go right into it, like I said, almost like cutting a fingernail to make sure you impact it less. you can see you've got some sand coming out of the drill right there. So he's trying to clean it out. Carl is standing underwater actually, so he's standing on the sea floor doing this, and he's using scuba equipment. And it took us a while to find this colony here. It took us about an hour because it is a larger on the larger side. There aren't that many large old coral colonies, meaning the larger the size, the older it is. And some of these coral colonies can actually live. Carl, how old do you think that coral colony is right there?
So the core we end up extracting from this particular colony, again, is I guess about 2 feet species.
So Carl's he's getting the drill ready. Again, it's really heavy underwater. How heavy is that drill, Carl?
Yeah, this part, again, this is what we call a hydraulic system. So it's driven by fluid that is running through the hoses that you actually see. A relatively heavy drill compared to the pneumatic system that I demonstrated earlier.
Yeah. So there it goes, and you'll see that you have some debris is coming out of the coring site, and that's to make initial contact with the skeleton. So you can see Carl's holding it vertically, yeah. So that is the exact process that this core came out of right here.
Awesome. So
and as you can see, the water's really green. Usually the water is much more blue. We had a lot of rain here, and there's a lot of things in the water that make the green. Some of that pollution coming in from the land, maybe that's impacting the growth of the coral as well.
So then after you extract that core, do you have the plug with you and put it in immediately, or do you come back later? What's that process like?
We put it in right away. And I think we have some pictures of the remediation pre and post. Remediation meaning like when we put the plug in, we're helping to remediate.
Let me pull those up.
Impact,
Give me one second.
To lessen the impact. And so the pre.
Is this the pre?
Yep, right there. Yep. Mm-hmm. That's actually a brain coral. So that's not the coral from yesterday, but that's a brain coral, right?
Yeah. Mm-hmm.
And that's from Florida. And you see we've got this grid, and the center square of that grid is, and then so that's where Carl is going to put the drill into. And I'm taking photographs because we're going to take a photograph before and after, and the grid helps us measure. If we come back a year or 2, 3 years later, if there's kind of any damage, we can measure that. We've got a ruler on the side to help with that measurement. So do you want to pull up the post photo, the photo from after the coring process? There it is. So, Carl, do you want to explain what's going on here?
Definitely. So the idea here is, so you could imagine that the cement is inserted, and now what we did is we went ahead and sealed the top of the coral colony, or where the core was extracted, with an underwater epoxy. This means that we have a combination of a glue-like medium that we use to seal off the top of the colony. The hope here is that eventually the coral will start to recolor area. And again, we're trying to track this to ensure that there's minimum damage in terms of, what we did in terms of extracting the coral core.
Right, because again, Sam, these were, the reason why we cored these really, really large colonies is because they're really old, and they can tell us a lot about the history. So if the coral is 100 years, the animal, that core can give us 100 years, been there for so long. So we really want to history to make sure, but damaging that. And also, it's really important.
Yeah, I think it's that, or to conduct this type of work, stringent permit process with the government of Panama, with the government of Belize, with Florida, in Florida as well, to ensure that we're tracking, how these corals are doing from year to year, and then we're required to report back to these government entities as well.
So what information are you looking at or looking for in these cores?
Okay.
Yeah, so, go ahead, Carl.
Go ahead.
Carl I'll start and Carl can continue. Like we said, looking in the past, we can these corals. So there's a photo I think we can bring up that actually shows what these cores look like when we take X-rays of them. So we actually take these to North Carolina, and we get X-rays, the type of X-ray called a CT scan. It's the same CT scan you would get if you were sick. And so it's kind of funny, we put these corals in the same place that these human patients are going. When we put them into the CT scan, we can kind of look inside and see that there are these bands. And these bands are the key to why these cores are so important. Because these bands are like the rings of a tree. So Sam, are you familiar with the rings of a tree and what they mean for a tree?
I am. I believe that tree rings represent rainfall. Is that correct? And so when you take a cross section or a piece of a tree trunk, you can tell the story of that tree by looking at the rings, and you can sort of learn a little bit about that tree's life history. So are you saying, Claire, that that's what you guys are doing with these coral cores as well?
Yes, so essentially you could imagine going back in terms, similar to what you just mentioned, you can imagine going back in time and looking at how the coral grew in a particular year and use that information to relate to environmental growth.
Like rain, yeah. Like
rain and whatever other additional environmental parameter the corals or the tree is exposed to. What's really cool here is that corals deposit what we call high and low density bands. So additionally, where forms are deposited. So paired bands, a high density band and a low density band are deposited every year. So we are able to go back in time and look at paired high and low density bands and use that information as some indication of how the corals grew in how a particular colony or coral grew in a particular year.
So Sam's bringing up these photos from like 5 years ago. So Carl, is that what you're talking about? The high and low density bands, the light and the dark there? Sam, is that clear to you?
I see vertical lines. Is that different in corals versus trees, where trees go outwards and these go downwards, almost like hamburgers and hot dogs?
So you could imagine if we look at the first image that is shown here, if you start from the very top of the image, you notice that the first band that is seen is a bright band that is a high density band. So that was deposited. Band, a dark band is deposited. So now we know what the first year of growth is, when the coral colony was collected. So if it was collected in 2008, that particular band, depending how late in the year it was collected, would be representative of growth the previous year in 2007. And then we would count back in time to get some indication of how the coral grew in, let's say, 2009, 2008, 2007, and so forth, as we count back in time.
So what Carl's saying is that see in that first one that's represented by the A, so every white layer you see is another year we're stepping back in time.
No, the white layer and the dark layer are representative of 1 year.
Right.
So an additional thing that I think is very interesting, Sam, is that we know that high density bands are actually deposited during the low density bands are deposited during the dry season here in Panama, which extends from December through March or so, mid-March. And we know that the high density bands are deposited during the wet season, which extends from mid. So we're able to March to November here in Panama. At least it's slightly different. The weather say, hey, let's figure out how this coral grew from year to year for a particular band. So sub-annual information can also be acquired from this as well.
Yeah. So what Carl's saying, Sam, is just like you were talking about how rainfall is really important to trees. It's really important. I mean, some environmental things like rain and heat are just so important to everything that lives in the ecosystem that you can see it literally, you know, inside of them, and a big influence on how they grow and eat and live. And that's why we can see them, you know, trapped in the coral and in the tree. And so that's why things like climate are just so important, because they influence everything about the animals around them, which I think is really cool.
I think so too as well.
So, you know, we've given you a lot of information. A lot of it's really complex. And this is only the start of a really exciting project for Carl and I. But maybe we can answer some questions about just kind of what it's like to sort of work. Sam,
Sure.
Questions coming in?
You must have read my mind, because we have a question from a student, and that question is, what do you like best about your job?
So, Sam, I think you broke up, but I think I heard you. Our job, I think part for me is contributing to coral reef conservation overall. That's what I think the primary goal is. This is why I'm doing this in the first place. The science is definitely important because it helps me to make recommendation to marine protected area managers as to how, you know, to conserve coral reef in climate change, and that is the part that is most important to me in terms of, you know, what I'm doing here.
Yeah, for me, I really like the exploration part of it. You know, climate change, or you know, the ocean getting, you know, it's really never happened this quickly before, and so there are things that are changing in the environment that are just, you know, we don't know about, and so there's so much to learn about the way the environment is changing because of climate change. And it's exciting to see what that is, and then it's exciting to report back, like Carl said, to you know, people in Washington who policies, to conservation managers, and even to people, you know, like you, Sam, working at National Geographic, who uses you know, photos and media to get the message out. And so for me, it's really the exploration part, which I really enjoy, because there's so much new stuff to still understand and to still uncover.
So another question. Why are corals a good species to use to study climate?
Well, the cool thing is that those bands have some idea of responding to environmental conditions in 1919, for example. So this can tell us what that relationship was, you know, in the early 1900s, and we can also use that to get some indication of how they are responding currently, and then how they are likely to respond to future changes as well.
Yeah, so I guess what Carl's saying is that, you know, to understand climate change, you need to look at things over really, really long time periods. You can't really just look at things like what happened in the weather today or yesterday. You really need to look at these long, like 100 years. So we really can't 100 years, and for 100 years. But because corals live for so long, and they have this permanent record in their schedule, like really unique. Secondly, the other reason why corals are such good animals to study for climate change is because they're really, really sensitive. They don't like it really, really hot, and they don't like it really, really cold. And so they visibly respond through coral bleaching or through death or through other things when the environment just isn't right for them. So they're kind of like it's kind of like a signal for us to monitor, to understand how fast the environment is changing, because they're just so sensitive.
So corals act like a big time capsule for the ocean almost, and have all these clues that we can use to document climate. That's really cool. All right.
That's exactly right. You put it perfectly, much better than we could put it.
I think so. I think great job there, Sam. But what's really cool, I like the way that you said that, is because, again, we can go back to a particular year and collect, you know, the skeletal material that was deposited. You'll be able to break the skeletal material that was deposited, and use that to reconstruct or try to get some sense of what the environmental conditions were, in a particular year. For example, we know that ocean acidification, we haven't spoken about that a lot so far, but ocean acidification is a major issue. So we can go back, collect collect that particular sample and use that to get some idea of what the pH of the ocean was, again in a particular year in 1919, and see what's going on. So that's also very important as well.
Yeah, so basically the time capsule is not only recording growth and temperature, but the chemistry too. So like, what are the chemicals in the ocean? When Carl's saying ocean acidification, that's just something that's happening where the chemistry of the ocean is changing. And so this time capsule is locking in information about weather, rainfall, and chemistry.
How do you know what band in the coral correlates to what year? How do you figure out specifically what part of the coral represents, like 1919? That was so long ago.
Yeah, it's a really tricky situation. You try to do it easily?
Sure. So essentially, you could imagine this is the core that we collected yesterday. So we know that the most recent band was probably deposited last year. So we'll define that as 2014 band, and then we count 2013, 2011, and so forth. So that gives us a good idea of when each paired bands were deposited from most recent time all the way to the deep past.
That is much simpler than I thought it was going to be. So you guys do all sorts of stuff. Even though you're marine biologists, you've got mechanical engineering with all of your tools, you've got chemistry with ocean acidification, and you have simple math with counting. That's fascinating.
Yeah, well, I think you need to have a really big toolbox as a scientist. And not to mention, so we have all the science, but we also need some of the practical skills, like, diving. Physically demanding. So I always stress to people that if you want to be a marine biologist, you need to be in good shape sometimes, right?
Definitely. Very important.
Yeah.
So I think we're going to start to wrap things up, but one more fun question. What did you both want to be when you were growing up?
So honestly, I knew pretty early on that I wanted to be a marine scientist. Again, I grew up off the coast of Belize, so I was out in the ocean pretty much every day. Our house is on the coast of Belize, and I was always excited about the ocean. I always wanted to see the organisms that live in the ocean, and as a result, I knew relatively soon from I was very young that I wanted to be a marine biologist, but not necessarily on corals, but I was just excited about all the organisms and the animals that I saw in the ocean.
Yeah. Sam, can you is it possible to bring up the map maker to show where Belize is for people who might not be familiar?
Yeah.
And so while you're bringing that up, I'll say that I have an essay that I wrote when I was in 5th grade that said I wanted to be a marine biologist. And so it's been the same for me. I grew up near the beach, not very much the Caribbean, but I still got a sense for, the way organisms live in the ocean and how things change. So that's, Carl, do you want to talk about Belize?
Yeah, so you could see Belize bordered to the north by Mexico, to the west and south by Guatemala, and of course to the east by the Caribbean Sea. So again, I grew up in southern Belize, all the way far to the south. Guatemala is about an hour away by boat, an area called Puerto Barrios in Guatemala as well.
Yeah. So we talk about time capsule. I mean, Carl's brain is kind of a time capsule. Since he grew up, he knows what those Belizean reefs looked like 30, 40 years ago, which is just so helpful for people like myself who just started exploring Caribbean coral reefs for the past 7 years. And having scientists like Carl who have a lot of that just information from firsthand experience. Imagine if your backyard was a coral reef. That was Carl's backyard growing up. And so he has a lot in just kind of like local knowledge coral reefs from his point of view, which is a very familiar and personal point of view, I think, that's different from other people.
So how does what you're doing in Panama and what you've done in Belize and the Bahamas and Miami, how does it all relate? What story are you guys trying to tell at the end of the day?
Yeah, so we initially started by conducting this work in Belize, and we're trying to get a sense of how corals overall on the Belize Barrier Reef system were responding to climate change. But again, the species we're interested in extends from beyond their southern range, which is again a little below Panama, and then all the way up to Florida. And we wanted to get a larger picture of how these different coral species were responding to climate change. So we decided to expand our work, to the northern range of the species, again, all the way up to Florida, and then to the southern range, which extends all the way down to Panama.
Yeah, I like to think of buffalo, for example, or bison in America. You can find them from Colorado all the way up through Alaska, and if you just studied the buffalo that were in Yellowstone, just in Wyoming and Colorado, you wouldn't really know everything about them, because you really need to study what the entire range of that animal is to understand how it eats and gets energy and lives, but also how it's reacting to the climate patterns that are changing across that range. So that's why it's so important, if you're interested in an organism or an animal and how it's responding to the changing environment, you need to understand it from the species that are in the south and the most north to get a really full picture. Does that make sense, Sam?
Yeah, so you have to look big to be able to see everything. So it's almost like if you're at a playground and you're on the ground, you can only see what you can see. But if you go up to the top of the swings, you can see much further and get a better view.
And if you are studying corals in Belize and there are some impacts or local impacts that are only specific to that area, you might not get a good representation of how they're responding because of those local impacts. So it's important to, again, scale up a little bit and get some sense of what's going on in Florida, in Belize, and in comprehensive representation of how corals are responding.
Yeah. So one last thing I'll mention is that Carl, a couple years ago, he found that there's some ways that corals are coping with increasing temperature, and hope for the way corals are going to might survive in the future. So is this coping, is their ability to kind of cope with the increasing temperature, is that unique to this tiny little swing set like you said, or is it happening all across the playground? Is it happening all across the Caribbean? So that's why we're going everywhere to understand whether this hope for coral reefs for the future is something we can expect from the entire Caribbean, or it's just this one little swing set of the playground, basically.
Great. So last question, and then we'll let you guys get back to your very busy day researching.
So do we have any more questions or photos maybe you want to show?
If you have more photos, just let me know, and I can pull it up while I ask you. If any of the students watching want to be scientists, what are some skills that they can start practicing now? What are some things they should do? How can they be scientists in their own lives today and scientists in the future?
Yeah, I think a great place to start is just going out and observing the environment. This is what scientists, all the way back used to do. They would go out, observe the environment, and see what is happening. So I think for science students and what's out there, this is going on. Definitely observing one way that a lot of that will come naturally and start. Just, of course, it's also important for them to get a good understanding.
If you want to pull up, there's a photo called of our study species, Sideras or Siderea. Maybe pull that up as a closing shot of the animal. Carl said, I wasn't always the best at science. I remember I got really bad grades in chemistry, and I didn't really like physics, biology, and I just asked really good questions. And I think the most important thing about being a scientist is just asking really good questions. Seeing, like Carl said, seeing something in the environment, like why do those frogs only live in that area, down the street? Maybe there's a reason for that. Well, what's the reason? Ask some really, really good questions is the most important place to start, I think. I want to just leave this photo here that I took. One of the coral species that we study here. It's actually the core. It's the type of coral that we cored in the video you saw. And one thing to take away here is that we're on this very important mission to get more knowledge. But at the end of the day, the environment is just so beautiful, and it's so brings so much wonder and excitement for me. And I love being in the environment too, and taking these wonderful people like you and people I interact. When you share a photo like this, you can just really see animals are and how beautiful they are and how much they mean to the diversity of life here in the ocean. So, anything else?
Yeah, again, this particular picture shows, that these corals, there are corals out there that, seem to be doing relatively well. And again, we're trying to get a sense for, how corals overall are responding. And, so far, the answers are quite interesting, and we're hoping to share this more and more with, like Claire said, policymakers, research managers, students, teachers, just the general public, for them to get a sense of why it's important for us to look at these questions and how climate change are actually impacting coral reefs.
And I want to thank Sam and Geographic Learning for having us today. It's been really fun to put together, ways to show you how we do what we do, and just to share, our interests with you folks, because it's so important to us.
Yep, definitely. Thanks, Sam.
Well, we want to thank you guys, Claire and Carl, for taking time out of your busy research schedule to talk to us about corals and the important work that you're doing. And we also really want to thank the Smithsonian for allowing us to, crash their research station for the morning. Thanks for sharing all your thoughts with us. Can't wait to have all these new kids become good observers and, more importantly, good communicators like you. We promise we'll post that video of the Smithsonian later. What?
If you want to follow Carl and I and our research team as we travel, through Panama, Belize, and we might even go to Cuba next year to continue this work, you can follow us on Twitter, and the handle is Castillo Corals, so it's CastilloCorals, and I'll be, I'm sure, on the Twitter feed. Or you can also follow us on Instagram using the same handle, Castillo Corals. So please follow us along and tweet at us and ask questions. We'll always respond.
So that's the best way to stay in touch with Claire and Carl and get the most recent updates on their research. And for the most recent updates on all of our explorers, follow @NatGeoLearning on Instagram. Thanks, everyone, for joining us today. It's been wonderful to learn and to visit sunny Panama. So thank you, and hope to see you next time.
Thanks, guys.
Thank you, Sam. Take care.
Take care.
Thanks, guys.